Imagine the ability to precisely edit the very building blocks of life, our DNA. This is the promise of CRISPR-Cas9, a revolutionary gene editing technology. CRISPR, short for clustered regularly interspaced short palindromic repeats, is like a molecular scalpel. The CRISPR-Cas9 system was discovered in bacteria, which use it as a defense mechanism against viruses. When a virus invades, the bacterium captures a snippet of the viral DNA and integrates it into its own genome. If the virus attacks again, the bacterium uses this stored DNA to guide the Cas9 enzyme to cut and disable the invading viral DNA. Scientists quickly recognized the potential of this bacterial defense system for gene editing. By providing Cas9 with a guide RNA molecule that matches a specific DNA sequence, researchers can direct the enzyme to cut DNA at any desired location. This ability to precisely target and edit DNA sequences has opened up unprecedented possibilities for genetic research and its applications. Just as bacteria use CRISPR to protect themselves, we are now harnessing this powerful tool to edit genes and potentially cure diseases. CRISPR-Cas9 works with an elegance that belies its profound implications. Picture a pair of molecular scissors guided by a tiny compass. The scissors are the Cas9 enzyme and the compass is the guide RNA. When the CRISPR-Cas9 complex is introduced into a cell, the guide RNA searches for its matching DNA sequence. Once it finds its target, the Cas9 enzyme snips the DNA at the precise location specified by the guide RNA. This break in the DNA strand triggers the cell's natural repair mechanisms. The cell has two primary ways to repair a broken DNA strand. The first is called non-homologous end joining, a quick-fix solution where the cell simply glues the broken ends back together. This method is prone to errors and can introduce small insertions or deletions in the DNA sequence. The second repair pathway is called homology-directed repair. In this pathway, the cell uses a similar or identical DNA sequence as a template to repair the break. By understanding and harnessing these natural processes, scientists can now make precise modifications to the genetic code with unprecedented ease and accuracy. The advent of CRISPR-Cas9 has been nothing short of transformative for genetic research. Its simplicity, precision, and versatility have made it an indispensable tool for scientists across various disciplines. Imagine being able to study the function of a specific gene by simply switching it off or introducing a precise mutation. With CRISPR-Cas9, this is now a reality. Researchers are using CRISPR-Cas9 to create animal models of human diseases, allowing them to study disease progression and test potential treatments. By introducing specific mutations into the genomes of mice, scientists can create models for diseases like cancer, Alzheimer's, and Parkinson's. These models provide invaluable insights into the underlying mechanisms of these diseases and accelerate the development of new therapies. CRISPR-Cas9 is also being used to study the function of genes in unprecedented detail. By systematically targeting and disrupting every gene in a cell, scientists can identify which genes are essential for specific cellular processes. Beyond its applications in biomedical research, CRISPR-Cas9 is also being used to develop diagnostic tools for various diseases. Section 4. A revolution in medicine. CRISPR's potential for disease treatment. The potential of CRISPR-Cas9 to revolutionize medicine is vast and incredibly exciting. Imagine a future where we can cure genetic diseases at their root by correcting the faulty genes that cause them. Clinical trials are already underway to test the safety and efficacy of CRISPR-Cas9 in treating a range of diseases. One promising area of research is in the treatment of blood disorders like sickle cell disease and beta thalassemia. In these trials, scientists are extracting blood stem cells from patients and using CRISPR-Cas9 to correct the faulty genes. The corrected stem cells are then transplanted back into the patient, where they can produce healthy red blood cells. Early results from these trials are promising, with some patients showing significant improvements in their symptoms. Section 5. CRISPR in agriculture. Cultivating a sustainable future. The applications of CRISPR-Cas9 extend far beyond the realm of medicine, reaching into the fields and farms that feed our planet. Imagine crops that are more resistant to pests and diseases, require less water and fertilizer, and produce higher yields. CRISPR-Cas9 is poised to revolutionize agriculture and enhance global food security. One of the most promising applications of CRISPR-Cas9 in agriculture is in developing disease-resistant crops. By using CRISPR-Cas9 to target and disable genes that make plants susceptible to diseases, scientists are developing crops that are naturally resistant to devastating plant pathogens. CRISPR-Cas9 is also being used to enhance the nutritional value of crops. Researchers are using CRISPR-Cas9 to increase the levels of vitamins, minerals, and antioxidants in crops, making them more nutritious and beneficial for human health. Furthermore, CRISPR-Cas9 is being used to develop crops that are more resilient to the effects of climate change. Section 6. Ethical Considerations: Navigating the Uncharted Waters of Gene Editing. The power of CRISPR-Cas9 to edit the very fabric of life raises profound ethical considerations. While the potential benefits of this technology are immense, it is crucial to proceed with caution and engage in thoughtful discussions about the ethical implications of gene editing. One of the most pressing ethical concerns is the potential for unintended consequences. While CRISPR-Cas9 is highly precise, it is not perfect, and there is a risk of off-target edits, where the enzyme cuts DNA at unintended locations. These off-target edits could have unforeseen consequences, potentially leading to harmful mutations. Another ethical concern is the potential for CRISPR-Cas9 to be used for non-therapeutic purposes, such as enhancing human capabilities. The use of CRISPR-Cas9 in germline editing, where edits are made to reproductive cells and passed down to future generations, raises additional ethical concerns. To address these ethical considerations, scientists, ethicists, policymakers, and the public need to engage in open and transparent dialogues about the responsible development and use of CRISPR-Cas9 technology. Section 7. CRISPR, a beacon of hope for the future. The discovery of CRISPR-Cas9 has ushered in a new era of genetic engineering, one with the potential to reshape medicine, agriculture, and our understanding of life itself. Its precision, versatility, and ease of use have made it an indispensable tool for scientists across various disciplines. The potential applications of CRISPR-Cas9 are vast and awe-inspiring. From curing genetic diseases to developing climate-resilient crops, CRISPR-Cas9 holds the promise of addressing some of the world's most pressing challenges. As with any powerful technology, it is crucial that we proceed with caution and engage in thoughtful discussions about the ethical implications of gene editing. By carefully considering the potential benefits and risks, we can harness the power of CRISPR-Cas9 to create a brighter and healthier future for all. The CRISPR-Cas9 revolution is still in its early stages, but its impact is already being felt across the globe.